or the parasite genome to see if we could see what does the parasite do. Unfortunately, we had too few reads. However, we wanted to see if the number of reads changed over time. And the way we are doing that is that we are normalizing the reads that aligned to count per million. And then we did a linear regression mapping the reads over time, allowing us to see differences for the time points in both fish species. So what we found generally were that zebrafish initiated an immediate response, peaking at around 24 hours with 1,231 differentially expressed genes. I do want to emphasize, however, that the majority of those were actually downregulated, and that very rapidly after this, the number of genes actually went down and the response normalized. The response for the rainbow trout were markedly different, with a very subdued response overall, with the majority being 28 differentially expressed genes at 24 hours. Overall, we also had a higher number of downregulated genes, but it was arriving at around 48 hours post-infection instead of 24, and still it's markedly subdued compared to the zebrafish, highlighting again a difference between a resistant and susceptible fish species in the early response. Because of the very few number of differentially expressed genes that we are actually getting for the rainbow trout, we could not do the enrichment analysis for those, where for the next responses here will only be zebrafish that would be presented. We did the enrichment at the gene ontology, and all the genes were then classified into either molecular function, which is the gray, biological process, which is the blue, the green, sorry, and then cellular components, which is the blue one. And then we looked into which one of those are immune associated, and we actually found that there were genes assigned to categories such as type 1 interferon production, immune receptor activity, response to cytokine, cytokine-mediated signaling, cellular response, and humoral immune responses. So it shows us here that there is initiating an immune response, both all the way from 2 hours and up to 48 hours. We also did the cake pathway analysis, where we were getting 8 significantly enriched pathways. 7 of those were at 2 hours post-infection, with the majority being associated with metabolism, 1 with cellular process, which was ferroptosis, and then 1 at 24 hours, which were the cytokine receptor interaction, linking very well to what we saw with the gene ontology and what we can also see through literature and through paper 1, where it's clearly showing that cytokines are being affected highly with an infection with EEC. Because we wanted to look into the immune response, and there were very little immune response related genes or pathways that were significantly enriched, we decided to go in and remove the cutoff. So we actually got all of the pathways that had differentially expressed genes assigned, and then went in and filtered those for everything that were immune related within the immune system category. And we found that there were differentially expressed genes assigned to 6 pathways: toll-like receptor, nut-like receptor, C-type lectin, Riga-like, cytostolic, and intestinal immune network for IgA. What I want to emphasize here is that despite having very limited number of genes assigned, all of them were more or less downregulated, indicating that the immune system were actually maybe being downregulated. I also have to say we only found immune system categories at 24 hours, where the majority of genes were also downregulated. The only one upregulated were CXCL11. This response is a bit different compared to other fish species, such as rainbow trout and Tibetan highland fish, where they see a high number of immune system categories being regulated. However, they are also looking at the late stages of infection and their susceptible fish species, highlighting again the difference in response. We looked at the number of reads that were mapping over time to see if we could detect whether the parasite were leaving the fish or if they were still present, or what happened with the development over time. And what we saw here with the blue box plot is that number of reads mapping to the the parasite genome is actually decreasing over time for the zebrafish, indicating that the infection is not progressing, that the parasite is being killed or cleared. Whereas for the rainbow trout, they're actually increasing very steadily over time, indicating a disease progression. We looked briefly into other genes that were also being assigned to categories that are not immune system but immune relevant, and especially the cytokine receptor interaction came up for us here, with a lot of genes assigned to interleukins, TGF beta, receptors, CD40, and so on, highlighting that there is a pitfall here in only looking at immune relevant, the immune system category. You also need to look a bit broader. We also looked at whether there were genes that were not assigned to any of these categories, but that might be relevant. And what we saw was that CXCL8A, CXCL13, and CXCL11 were actually significantly upregulated. And that's rather interesting because they're actually all associated with the cellular response, highlighting again the difference and the importance of these. So with this transcriptomic study, there were some differences in the experiment that might influence the results. So we compared a tropic fish species to a cold water fish species. And since immune responses are influenced by temperature, this might have influenced the result. However, we decided to run the experiment with optimal conditions for each species because we wanted to be sure that what the response we were seeing were not a matter of fish that were living or going in not optimal conditions. We wanted to be sure that everything was optimal so we could trust the results that we were getting. There's also a surface to volume ratio that are different between big rainbow trout and small zebrafish, especially because the parasite are not really different in size depending on who they're infecting. So they might do more damage or be more irritating for zebrafish compared to rainbow trout. There's also a difference in the isolates. We did them with similar abiotic factors at what they would normally go with. However, there's still difference in the isolate in terms of virulence, mortality, serotype, etc., and that might also influence the results a bit. We would have liked to repeat the experiment to show that even though you get a different isolate or maybe try and mitigate these abiotic factors, we will still see the same results. However, we were not possible to do due to economy finances. So based on the findings from papers 1 and papers 2, along with the literature search, we found that it appeared as the cellular response were very, very important for the resistance of zebrafish toward the disease. And then we looked into what were actually upregulated and what were regulated across literature, and found CXCL8A as a candidate. The reason why this was a candidate was because especially neutrophils were very active in killing or associated with killing the parasite. And this particular gene is responsible for attracting neutrophils to the site of infection or injury or damage, meaning that it's very important for the neutrophils in terms of getting to the right spot and attacking the parasite. And therefore we decided to see if this could be a potential resistance gene. And we did that by creating a knockdown or a knockout of CXCL8A in zebrafish by creating a 289 base pair deletion using the top 2 guides. Additionally to that, we also had a scrambled control group, which is a control group that had injection the similar way as our knockdowns did. However, we injected with a guide that does not bind anywhere in the genome. And then we had non-injected controls. We infected them with roughly 300 therons per larvae, and then we counted the number of established white spot at 24 hours. After we'd done that, all of the larvae were placed individually in individual PCR tubes. We incubated them at 95 degrees for 30 minutes before we ran a PCR, allowing us to identify which fish were mutated or which larvae were mutated based on the appearance of fragments. So the top fragment up here is the wild type band, which is roughly around 472 base pairs. We expected that to be present in the majority despite being mutated, because CRISPR often only attacks one allele and not both. And then we could see the mutated would have the second band in the bottom, which is around 143 base pairs, because we've managed to take these 289 base pairs out. So we had a 62.5% deletion rate, successful deletion rate, meaning we had roughly 30 larvae that had a mutation out of the 48. And what we found was that when taking the number of successfully infected therons, or white spots, the mean number were significantly higher in the knockdowns compared to both scrambled and non-injected. There was no significant difference between scrambled and non-injected. However, the mean number were a bit higher, probably reflecting that when you inject things into them, the fish becomes a bit weaker. So what we hypothesize is happening is that once we create this knockdown of CXCL8A, neutrophils are not being attracted to the parasite, allowing it to develop this cytostome, so its mouth, which then makes it possible for it to start eating potential immune cells and other cells, so it can grow even bigger, coming to a point where there's a point of no return, where the fish can't do anything against the parasite anymore because the parasite becomes too strong. Now, I do also have to express that CXCL8A is expressed by leukocytes, and when there is an infection, there is a natural attraction of leukocytes to the site of infection, meaning that it will increase, and the significant upregulation of CXCL8A we see is potentially it's more a reflection of an increased number of leukocytes compared to an actual upregulation of the gene. One way to see that would be to do cell sorting and look at what does the cells express. Do they express more, or is it just an accumulation? Or to do spatial transcriptomics looking into these things. So generally and overall, what we found through these studies was that zebrafish larvae relies on the innate immune response, and when doing that, they mount an effective defense against ick, which involve especially neutrophils and macrophages. And we suspect that the key to a successful defense lies in the early initiation, preferably within hours of infection, preventing the parasite from being able to start ingesting immune cells. We found that the resistant zebrafish initiated an intensive early response, whereas for the susceptible rainbow trout, it was very limited. And that might be due to the fact that the rainbow trout, maybe it doesn't acknowledge the parasite as a threat until it's too late, because they're so small, or maybe it's the fact that the parasite manages to do immune evasive behavior where it's hiding itself from the immune response, and then it's again allowing it to develop the cytostome, growing up and becoming big. While the majority of the immune-related genes were significantly downregulated, various cytokines were upregulated, and these cytokines were associated especially with the cellular response, where for our study, especially CXCL8A were interesting due to its associating with immune cells capable of killing the parasite. And we found it upregulated in both larvae and adult zebrafish, and we found it upregulated across studies, allowing us to hypothesize it as a resistance gene. And then creating a knockdown of CXCL8A in zebrafish resulted in a significantly increased parasite birding, demonstrating the functional relevance of CXCL8A, but how it affects the cells need to be investigated further, but at least in terms of these studies, indicating that it is a resistance gene for zebrafish. I would just like to briefly acknowledge my supervisor and co-supervisor, Louise and Morten, from the University of Copenhagen, and our collaborators from the University of Sheffield, Professor Stephen Renshaw and Catherine Loyens, for their assistance with the whole CRISPR work and teaching me how to do it, designing guides and everything. And then, of course, the funding bodies, the Danish Research Council. And yeah, to all of you, thank you very much for your attention.
Okay. Thank you, Mrs. Heidi. Okay. What a great and insightful presentation. Okay. And now we move to the next agenda. I would like to invite Mrs. Huria Marnis, PhD, to begin the presentation. You have 35 minutes for your presentation, Mrs. Huria, and the screen is yours.
Okay, thank you, Mbak Anissa. Let me share my slides first. Right. Okay. Is it visible on my slide, Mbak Anissa?
Yes, yes.
Okay, thank you. All right. Yes. Okay, thank you so much, Mbak Anissa, for the time. And first of all, I would like to say thank you very much for our head research organization, Ibu Dr. Puji Lestari. And also I would like to say thank you very much for our head research center, Bapak Dr. Fahrozi, for giving me an opportunity to share our knowledge in this webinar. And then, hi, Heidi. I would like to say thank you very much for your presentation. It was really nice, and I'm glad to see you again, even though in virtual meeting. Thank you. And then good afternoon, everyone in Indonesia. Good morning in Denmark time. I hope you are in good health and get more valuable information in this webinar. Okay, today I want to share about transcriptomic analysis of Baltic cod liver infected with Contrasocum oscillatum Thursday larvae indicates parasitic effect on growth and immune response. Actually, this is part of my PhD project, and this is a painting from my supervisor, Kurt Bertman. I would like to say thank you so much for him for providing this painting, actually, and this painting also related with this study. Okay, actually, this study already published a couple years ago. If you want to know more detail about this, please feel free to contact me. And then let's start with the background of this study. This is a picture of Baltic cod. As we know that Baltic cod is a substock of the Atlantic cod. In Latin name, we call it the Gadus marhua. This species inhabits the Baltic Sea, and of course, in economic commodity and high focus is placed on quality and health of the stock. And this is the map distribution of the cod. It distributes around North Europe and North of America. Several subpopulations seeking deeper water for spawning. So when we look a map of Baltic Sea, there are genetically spread stock of cod in Baltic Sea area, in the eastern area and the western, and mixed zone around the bottom area. And then during recent years, marked increase of the occurrence of Contrasocum oscillatum Thursday larvae in cod liver has been documented. A lot of report say about that. And this is a picture of the Contrasocum oscillatum Thursday larvae after we collect from the cod liver. So when we zoom it, it looks like this. And then the rise of the infection has occurred at the same time as the Eastern Baltic cod stock has faced historical crisis. It is therefore relevant to analyze the connection between the parasitism and inferior performance of the cod. And then this is how the parasite look like under electron microscopes. So this is part of the posterior in the and this is interior part, posterior part. We can see mold here in the parasites. And also when we look more detail, so this is boring teeth. And this also part of the posterior. And the adult stage of the Contrasecum oscillatum was originally described by Ascaris osculata by Rodolfi. This is a painting of Rodolfi, painted by supervisor again, Kurt Buchmann. And this parasite actually recovered from the stomach of the seal in the western of the Baltic Sea. And the parasite Contrasecum oscillatum has been subdivided into several sibling species, including 3 Arctic species, and including Contrasecum oscillatum B, A, and C, and then 2 Arctic species like Contrasecum D and E. For the Thursday larvae of the Contrasecum oscillatum, sensus tricho or type C has been found in the cod, spread, and gray seal, identified using the mitochondrial gene like COX1 and COX2 by our college in 2016. And then this is the life cycle for the parasites. So the gray seal or mammal has a final host for the parasite, and cod as intermediate host. So let's start from the gray seal. Actually, feeble worm adult stay longer in the stomach of the gray seal, and their age reach the environments through with the gray seal phases. And then age being Thursday larvae, this is Thursday larvae, and crustacean ingest the Thursday larvae, and then of course the small fish get infection because ingest the crustacean already infected with the these parasites. And again, a cod ingest the small fish already get infection with the parasite, and this parasite go through the gray seal body because a gray seal ingest a cod already get infected in the liver, and the adult parasite mold twice in the gray seal stomach, and yes, being adult in the stomach of the gray seal. And this is the life cycle for the parasites. And then liver is the target for the parasite, for these parasites. As we know that liver, fish liver, comparatively large organ supply both venous and arterial blood. And yes, this liver critical for production of protein, metabolism, and nutrient, of course, the clearance the toxin. And liver is also frontline organ of innate immune system. And then the Baltic cod liver is generally most heavily infected organs with the Contrasecum oscillatum Thursday larvae. According to the our college study, he reported that histological sections show that cell, fibroblasts, and macrophages of the cod liver encapsulated in the larvae. And then, like inflammation is induced in the liver during an infection with the Contrasecum oscillatum Thursday larvae. And previous studies saw that key PCR analysis has been performed using zebrafish and also rainbow trouts. The involvement of the gene in the inflammation associated with the infection. Of course, the result so that the overall expression of the immune gene test was downregulated after exposed to expression and secretion product of the parasite, including interleukin beta, interleukin X, TNF alpha, MHC2, CD4, and IgM. So to gain better understanding of molecular mechanisms underlying the host response to the parasites, we performed transcriptomic analysis. So this study, the aim of the study was identified and characterized gene expression of liver in the Baltic cods infected with the Contrasecum oscillatum Thursday larvae. All right, we move to the materials and methods. So we collect 10 samples from the Bornholm area. This is for infected group. And then we also collected samples from the blue planet. Actually, we count on the coastline of Zealand. This is for a control group or unaffected group. So we did selection based on performed based on parasite screening result, and then we took the liver from all of those our fish, and then we extract the total RNA either for control or for infected one. And after we get the total RNA, of course, we run the transcriptomic analysis using next generation sequencing, disease flowchart reference RNA sequencing workflow. First, of course, we do library sequencing, and then we perform transcriptomic sequencing, and then we align our sequence with the Gadus Marhua reference genome, and we perform differential expression analysis. Of course, we did visualization and data exploration analysis, and yes, we perform validation of transcription data. All right, and for the gene ontology annotation and KGG pathway analysis, we use David software. Okay, and we did RNA sequencing data were validated by a key PCR. First, yes, we extract the total RNA and we perform the cDNA. Of course, we run the key PCR, and then so we select 14 for validation, and then we use for reference gene for validate RNA sequence. Sorry, key PCR analysis, and this is all gene that we use either for target gene or reference genes. And then this is the result for our study. Actually, number of Contrasecum oscillatum larvae in Baltic liver, we found around 100 prevalence of parasites, Contrasecum oscillatum Thursday larvae, infection in Baltic cod from the east coast of the Bornholm. This is for infected group, and infection level was positively correlated to fish liver size. And yes, no parasite or no Contrasecum oscillatum larvae were found in non-infected group. And both of our two groups, we found low infection of the trematode in caloric cecum lumen and the enterocephalan in the intestinal lumen. When we look more detail about the data, so we divide weight of the Baltic cod liver into 3 categories. The light one around 20 to 40, and then the middle one 40 to 60, and the wet ones around 60 to 80. So more, I mean, the weight of the vigor of the Baltic cod liver, so the most of the parasite that we found. And these were aligned to the Gadus Marhua reference genome. This is the link for reference genome that we use. And this is raw read account and map read gain reference. This is for control group, and this is for infected one. So the average raw read count we found around 17,000. And then for map read again reference, we found around 10,000. In total, around 60% using Top Hat software. And yes, we have deposited all those data to NCBI database. This is the accession number for this for those reads. And then for sequencing and annotation of prediction protein, we use the protein database from the zebrafish, because this is a zebrafish is fish model already established for protein databases, right? And we found around 47,000 unigene and around 12 unigene, or around 25% showing similarity to zebrafish proteome. And then we found around 34,000 unigene, or around 74% unknown protein. So that means it's potential for novel gene. And for differentially expressed gene, around 47,000 unit gene. So we found around 4.43% or around 2,000 genes were regulated. And when we look more detail, so we found around 40% genes or around 844 were down-regulated, and around 1,200 or around 60% genes were down-regulated. This is based on the cut-off significance was set to p-value and at least for two-fold things, and the rest were not changed. And then how it looked like when we plot in the Pocono plot. So we can see clearly here. So yes, around 45% genes down-regulate, and then around 1,200 or around 60% genes up regulation, and the rest in the middle were not changed. And then for gene ontology, so gene ontology is a classification generally assigns the functions of the gene and their products in organism. So we divide into three categories: cellular component, molecular function, and biological process. We found around 1,300 out of 2,000 active DAGs were assigned to gene ontology of function group. So some genes here were involved in the metabolic process and also a growth. So we focus on that. And then when we look more detail, so we found around 22 enriched GO term according with their exact p-value, and enriched term from a biological process include metabolic process here and also growth. And then for KGG analysis or KGG pathway, so we got around 454 DAGs were annotated to 168 pathways. And you know that KGG's database used to map associate genes to their respective pathways, which are classified into 6 categories here: human disease, organismal systems, cellular process, environmental information processing, genetic information processing, and metabolism. Yes, some of them involved in a metabolic pathway here. So we can see in this list of the enrichment KGG pathway in DAGs. And then we also found around 30 and 31 and also 32 genes were related to the carbohydrate metabolism. This are around 30 here. And then 35 were related to the amino acid metabolism. And then 32 genes were related to the lipid metabolism. Okay, for differential expression of immune-related gene in multi-code liver, we found around 13 genes were related to the immune function, including chemokine. So yes, some genes also relate with the chemokine here. And then yes, for differential expression of growth-related gene in the multi-code liver, so we found around 39 were differentially expressed in multi-code liver infected with the parasites. Several genes involved in key growths were down-regulated. So we can see here, keratin 91 was down-regulated, and then also IgM, like insulin-like growth factor binding protein, is also one key of the growth. Is also were down-regulated. And then for RNA sequencing validation using real-time PCR, so we can see here all the selected gene were encoding like C3, CRPP1, CRPP2, and IgD were downregulated. So we can see here downregulated. And then RNA sequencing results were mainly consistent with the real-time PCR result. And this indicating that the gene expression profile generated by RNA sequencing were reliable, and thereby confirming the observed change in the expression of the gene responsive to Contrasacum oscillatum torstic larvae. And for the conclusion for the study, so the infection of the parasite of this parasite, Contrasacum oscillatum, was associated with depression of some gene related to metabolism, immune system, and also growth. And infected fish need to invest energy when coping with the inviting pathogens, which indirectly may influence the growth of the host. This host is Baltico, right? And then it cannot be excluded that the parasite or parasite product directly regulate gene expression of the growth and immune-related gene. Before I end up my presentation, I would like to say thank you so much for my supervisor, Kurt Buchman, and my co-supervisor, Perukania, and our collaborator, Rondix from Future Genomic and Technology. I would like to say thank you also for the funding European Fisheries Fund and Danish Fisheries Agency and GPA Epperson and Mrs. Daphne Epperson Foundation. And yes, many thanks to my college for their support during my study in Copenhagen University. I think that's all. Thank you very much for your attention. Unmute, Mak Nisa.
Okay. And now we move to the Q&A session. Okay, if anyone wants to ask our speakers directly, please use the raise hand feature. Okay, we have one participant who raised hand. Mr. Hairul Syahputra. Okay. I think it is reunion. Okay, please, Mr. Hairul, for your question.
Okay, thank you. Good to see you again, Heidi. So we are so
You
happy
too,
to have,
Hairul.
yes, we are so happy to have you in this webinar. Thank you very much for your contribution. Yes. Yes, yes, and thank you very much for your presentation. It was very good, and then it's very nice. I mean, the quality of your project is I know it's very much work there. You have done so lot. Okay, I only have a question, Heidi. Maybe this just to encourage others to ask to involve this in this discussion. Actually, I was waiting. I mean, you study about the effect of the temperature on this parasite and also this fish, because of we know that temperatures affects both. I mean, to the development of the parasite and also the immunity. So I saw on your slide that you also compare the difference between, but you only do it. I mean, did it separately, right? Because I know it's the story behind, because you use a zebrafish because it's a tropical species, because you cannot do it a bit lower because it's not good for the fish, of course. And also you also did with the trout, because this is a cold species, right? But actually, I want to ask you about what exactly the effect of the temperature on the immunity, because we know we have there are innate and adaptive immunity. What exactly, does the temperature affect mainly to the innate or adaptive, or vice versa? That's only simple question, Heidi. Thank you very much.
You're welcome, and good to see you again as well, Harold. So it influences both the innate and the adaptive. From my understanding, it influences the adaptive a bit more actually compared to the innate. So in terms of this experiment, it's still fine. Or it's not fine, of course. It would be ideal if we could have gotten the fish used to the temperature, but then again, you would never really know, is it actually a response because we change things, or is it the parasite in itself? And that was always the debate back and forth. So there will be some differences. In the colder fish, there might be a little bit less response, so they might be more active immune response in the more tropical, because temperature does influence how many genes are expressed and how expressed they are. So that might be one of the things that have influenced. We did, however, go in and then estimated the degree days to try and mitigate the temperature. And the degree days showed that the sampling at 24 hours and 48 hours for the zebrafish and rainbow trout respectively were actually, I think it was 51 and 52 degree days, so very close. And what we saw in the response was still similar. So a big response in the zebrafish and a very small in the rainbow trout. So it's the best way we could mitigate the temperature thing, but it has influenced. Yes, it has, no doubt.
Yes, thank you.
Pak Hairul for the question, and thank you, Mrs. Heidi, for the answer. And now, is there anyone who raised hand again? Oh, not yet. Okay, we move to the chat. Mrs. Heidi, there is some question for you. The question is from Mr. Rahman Shah. Mr. Rahman Shah, question for Dr. Heidi Mathiesen. The first question, disease outcomes are determined not only by the parasite, but also by environmental conditions. How do environmental stressors, such as temperature fluctuations, hypoxia, and salinity changes alter host transcriptomic responses to parasitic infections? And the second question, future aquaculture should detect disease before clinical symptoms appear. Can transcriptomic biomarkers be developed into reliable early warning systems capable of detecting parasitic infections before clinical signs become evident? I think you can answer two questions first.
Yeah,
Okay,
so
go
in
ahead,
terms of the
please.
first questions, with the environmental stresses and how they influence, so they influence both the parasite, and they influences the response for the fish. Because if you have too high temperatures for a rainbow trout, it will be more weak, and then it will be more susceptible to the infection. But again, there's also a limit to what the parasite, how it can thrive. Because one of the ways that you can, at least in the ornamental pet shops, one of the ways that you can mitigate this disease is actually by increasing the temperature, because then it becomes unfavorable for the parasite. But generally, for the stressors is that they will weaken the fish. But they will also affect the transcriptomic response in terms of upregulations and so on, similar to what I said to Harold's question before. So there is definitely something there, and that's why it will be optimal to try and mitigate this. And that was one of the challenges by comparing a tropical fish to a cold water fish. So question number 2, I believe the transcriptomic biomarkers can be developed into reliable warning systems. And I completely agree that it would be very, very nice if we were able to detect this before it becomes a problem. I do think there is a different side to this one. So not only transcriptomic biomarkers, because the problem right now is that you need to sample the fish in order to do these, and then you need to catch the fish that are infected. If you have a very little infection, you might have parasites only in a few. So I think we can actually win more if we went the environmental RNA or environmental DNA way instead, because in that way you would get what is in the water, which will be reflected broadly, no matter which fish infected, and you don't need to take out fish samples to see what happens. But yeah, I do believe that transcriptomic biomarkers could be a nice warning system. Yeah.
Okay. And the third question, it is a great question, I think. A recurring challenge is bridging the gap between laboratory discoveries and commercial application. What are the major barriers preventing transcriptomic discoveries from being translated into affordable technologies for commercial aquaculture?
Oh, that's a very good question. Because I completely agree that one of the challenges is bridging this gap between laboratory and commercial. And one of the things is time. So from when you take a sample until you get the results, it might be too long. So that's one of the things. It's also a matter of price-wise. So it can be too expensive compared to the results you're gaining from it. Because again, if you take a sample from a fish, it will only show you that specific second. It's not showing you how things are developing, and that might also be one of the things. So we need something that's something that can potentially be done on the farm, and something that will give you a very, very quick response, because if it takes days for you to get a response, it might be too late in terms of being able to mitigate it, so that when you take your samples, it's always already potentially too late in terms of initiating a response to it. And I think that's one of the major things. So we need something that will give us a reliable result very, very early, that's easy to use, and just generally, yeah, more applicable on site. And I know it's a lot to ask for, trust me, I know. But from a aquaculture employee now, where I'm actually breaching this gap, is what we need.
Thank you, Mrs. Heidi. And the next question is for Mrs. Huria. The question is from Pak Nurfianto. The question is, good afternoon. I want to ask Mrs. Huria, has the research you have done produced a specific marker that can be used in fish breeding? Thank you. Ms. Huria, please. Yeah. Okay.
Thank you. Thank you, Pak Nur, for the question. A nice question, actually. Yes, our transcriptomic study has not yet produced validated a specific gene marker for use in multi-code breeding, actually. However, the analysis identified several candidate gene and immune and growth-related pathway that respond to Contrasecum osculatum infection. And these findings could provide the basis for developing a marker associated with the parasite resistance or tolerance in the future, right? And further validation using large core population, of course, and SNP genotyping, and then association analysis could be needed before any candidate marker could be applied in the breeding programs. And as we know that multi-code is, this normally we catch from the wild or nature. So because this code I think a bit difficult to cultivate, because its character need seeking deeper water for spawning. So this is also difficult for cultivate for a code. A multi-codes. I think that's all, Mbak Anissa. Thank you.
Okay, thank you, Ms. Huria. And the next question is from Admi Atira. The questions is for Mrs. Huria. And the question is, thank you for the clear presentation, Dr. Heidi and Dr. Huria. Appreciate to all of you. And this question is for Dr. Huria. I am very interested in your screening process. Could you please elaborate on what specific criteria methods were used during the parasite screening to confidently classify the fish into the control and infected groups? Thank you. Please, Mrs. Surya.
All right, thank you, Mbak Asmi. Yes, Bu Admi, for the question. Yes, actually, first we have to screen our fish free from parasite, right? So yes, because we coach from different area, so for all fish we have to check everything. I mean, we have to make sure the parasite free from pathogen for control. And of course, we check the liver and inner organ, everything inside of the fish, and so we make sure the fish is free from pathogen. And then, of course, yes, we found in our study we found some parasites, but not nematodes. In also for both infected or non-infected one, but it's not this still a low infection. So yeah, we have to screen our fish from the parasite. So just make sure everything is free from parasite, especially for a control group. Thank you, Mbak Anissa.
Okay, thank you, Ms. Surya. Is there anyone who can ask directly again? You can raise your hand. Oh, okay. I think it is my question, if any. Okay. I think I want to know what are the limitations of relying on transcriptomic data alone to understand host-parasite interactions? What additional experiments would strengthen the conclusion? Okay, Miss Heidi first.
But I think there's different things. So one of the things that limits is that when you do, for example, the enrichment analysis, you're doing it based on human data. And there are several genes that are homologous. There's also orthologs. There are different orthologs. So you might be risking to exclude information when you have to rely on something that's not made. Additionally, there's also, at least for rainbow trout, it's not very well annotated, the genome, so you're also losing a lot of information in that sense. The zebrafish is further along, so that helps, but still you're relying on things that are not one-to-one. So that might be a limitation as well. And also because it takes time to get knowledge, and you get so much knowledge that for you to be able to actually go through it and gain a full picture, but that's actually also a bit tricky because you can spend years on still learning things. And that's of course a good thing because you can learn a lot, but it's also in terms of getting it out there, it becomes a bit heavy. So yeah, I think that's some of the things that are challenging it. Because on the plus side, you also get a lot of knowledge from a potential single sampling compared to if you do qPCR, where you go and look for specific things. Here you get the broader, and you get knowledge on how things work together. But then there's also the factors that influences your experiments as well, that also can influence your transcriptomic analysis. The factors that we talked about, for at least for my experiments, but also when you do your alignment and everything, you risk also losing some there. So there's just so much filtering and everything that can also mask some of the responses a bit.
Okay.
I think that was
Okay,
my.
Okay, thank you. And Miss Surya, what about you?
I think same as Heidi, but I want to add some more. I think, yes, we have to consider about the number of the sample. So the minimum for the transcriptomic sample should be around 10, I think, like biological replicate. I mean, 10 for like a control, or maybe 10 for infected one, if you infected sample, right? And then, of course, reference gene. Reference gene is also important. So, and I think it's better when you use rake sequencing rather than de novo sequencing, right? So if your sample or your fish target has reference genome, I think it could be better when you use a reference genome. And then, so I think the integrity of RNA sample is also important, because you need to get better quality, the integrity of RNA sample around 7 to 10. I think it's better for you. And then, of course, the threshold of the raw data. So you have to careful about that. And then, yes, I think the important things. I think it's better for you to use control experiment. So you can get exactly, I mean, a real the transcriptomic, whole transcriptomic for the target organ that you want. I think that's all, Mbak Anissa. Thank you.
Okay. Thank you, Mrs. Surya, for the answer. Is there anyone who wants to ask again? We have only 2 minutes more.
What about the question from Johannes, Bu Anissa?
Oh, where's the question? Wait. Is this there is a question more in chat room from Dr. Rahman, from Mr. Rahman Shah. Question for Dr. Huria. How transferable are these transcriptomic findings from Baltic code to economically important tropical aquaculture species, particularly under tropical environmental conditions?
Okay. Thank you. Thank you for Pak Rahman for the question. It is a really nice question. Actually, yes, we know that the Baltic code is temperate. I mean, inhabiting temperate temperature, right? So I think so. So let me read the question again. So, how the this transcriptomic finding from the Baltic code economic important? Okay. Okay. All right. So yes. I think yes. I think because in this study we performed the transcriptomic analysis. Of course, I think we can apply this method as well for the another fish species. So we can use this as a model fish for aquaculture. Even though Baltic code is inhabits in temperate condition, so we can use this data as a model, right? And model because we can apply these techniques also for the first for the another species aquaculture in Indonesia. So we just need to the reference gene, the reference genome, and also we need a kind of specific fish that you want to study with. I think that's all. Thank you.
Okay, thank you, Ms. Huria. I'm sorry, it is the last question from Ms. Heidi. Okay, it is from Johannes. Hi, Heidi. Very interesting work you've done there. If I'm not mistaken, your data shows zebrafish resistance relies on a very early transcriptional window, 2 to 24 HPAI. If that timing is truly critical, does that mean immune priming strategies, like for example feed-based immunostimulant, have a realistic chance of improving resistance against itch, especially in other fish like trout or catfish? Would love to see your view on this.
Oh, well, that's a good question. Thank you very much for it. So in terms of if it can make a difference, I do believe it can, because you can, if you have the right feed additives that are effective, you can have stronger fish. And stronger fish can sustain the infection better and they will be more healthy. So in that sense, I do believe that it can make a difference. I don't think it necessarily make a difference specifically to the infection, but it does it in the, I'd say, the opposite way, where it's ensuring we have healthier fish, stronger fish, and more resistant generally because they're healthier. And I think in that sense, feed additives can make a difference. But in terms of making the fish able to expel the parasite, might be a bit more challenging. But again, if you have fish that are healthier, the cellular response will also be stronger. But I think it will be more difficult for the rainbow trout because there's still so much knowledge we need to gain in terms of why is it not acknowledging the threat in the beginning. So is it because the parasite are hiding itself, so it modulates the immune system? And if it does that, then maybe having a stronger immune response through feed additives will allow it to respond better. So I think there's potential, but there's just so much knowledge we need to gain in the response of the rainbow trout before we can set in efficiently with stuff such as feed additives and everything. But I will say that, yeah, stronger, healthier fish will do better in terms of mitigating it. Thank you.
Okay. Thank you, Mrs. Heidi. Unfortunately, we have run out of time for more questions. Thank you, everyone, for your active participation. And as a final conclusion, we learned that it is important for us understanding host-parasite interactions at the molecular level to improve fish health and disease management. Thank you once again to our speakers, Mrs. Heidi and Mrs. Huria, and to all participants for joining us today. We hope this webinar provide valuable insights and inspire further discussion or any collaborations in advancing fish health research and sustainable fisheries worldwide. Thank you. Have a wonderful day.
Thank you. And thank you, Heidi.
Thank you very much for the invitation and for the great program and everything. It's been a pleasure.
Okay, thank you very much, Ms. Anissa, and our distinguished speakers for the insightful presentation and valuable discussion. Ladies and gentlemen, we will now proceed to the certificate presentation session as a token of appreciation for our distinguished speakers and moderator for their valuable contribution to today's webinar. The certificates are presented to Heidi Mathisen, PhD, Huria Marnis, PhD, Anissa Fitri Larasagita, SSE, MSE. Thank you very much to all speakers and moderator for sharing your knowledge and contributing to this meaningful discussion. Ladies and gentlemen, we have now reached the end of the international webinar Aqua Talk number 8. On behalf of the organizing committee, we would like to express our sincere appreciation to all speakers, moderator, and participants for your valuable contributions and active participation. We hope that today's webinar has been provided meaningful insights and strengthened collaboration in the fields of fish health, parasitology, immunology, and sustainable aquaculture research. Thank you very much for your attention. See you at the next aquaculture webinar. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you, Ms. Heidi.
Terima kasih, Bu Andalusia.
Thank you very much, Heidi. Take care. Bye-bye.
You too. Bye.